Today I just want to present our recent work on developing a multiscale model that captures stress relaxation and flow in proliferating tissues with an emphasis on the subcellular elasticity. So, So here's like a quick overview of what I'll cover today. So I will start with like some of the biological background and motivation, and then I will introduce our vertex modeling framework, followed up by its application like the wound closure and wing growth. And then finally, I will talk about like our direction to our future work. So epithelial tissues are not like passive materials. They are active dynamic systems where like cells generate like internal forces, primarily through like ectomyosin contractility, cell-cell adhesion, remodeling, and turnover of structural components. And then these tissues, they exhibit like viscoelastic behaviors. So the elastic-like response is like when the tissue deforms quickly and the tissue store mechanical energy or the stress due to like cytoskeleton tension and cell junction elasticity. And for the viscous-like flow, it's like over long time scale. It's like the junction remodeling, like for example, the T1 transition, and then that's gonna relax the stress. This is what we define as the tissue fluidization. But a key question still remains, like how do like, the tissue simultaneously grow while like, maintaining their mechanical coherence? So this is the question that we want to find the answer. So most previous studies view these epithelial tissues as cohesive media connecting cells by intercellular junctions, like those catalin and actin, while treating these mesenchymal cells as individual particles. However, in development and cancer, cells often adopt a hybrid identity, which is called the PEMT here. Is where cells retains some adhesion and but also like gain motility. And then this like kind of motivates us to know more about this fluidity, because like how like their fluidity is organized and controlled still remain largely unknown. And so we kind of need like a new modeling framework to study this interplay between actin structure, fluidity, and morphogenesis. So here I would like to ground our modeling in large-scale planar tissue flow during wound closure. So I have the video here. So on the left video here is the monolayer wound closure on MEC1, which is the mouse embryonic epicardinal cells. And we also have the TGF-beta here on the cell as well. You can see like the middle part here, the space here is representing the wound area. And the cell or the tissue around there is trying to close that gap. So on the front, some cells are crawling, and some cells also have like proliferating or divisions. And some interesting behavior is that some of the cell is actually not moving, but it's not moving like into the area. It's actually moving outward. So that's what we call like the wiggles or the buckling effect. And compared to like the right video here, which is a different cell line, the MBCK1, you can see like the closing process, like the actual wound boundary is closing pretty like it's not smooth closing, and there's some like buffering effects inside the tissue as well. So this so basically our goal is to try to use our model to simulate such closure events. We're trying to use like tissue scale dynamics like from a subcellular forces in order to like mimic these events. So for the modeling framework, we are going to employ what's it called, the 2D vertex model. So basically, we're going to define like the tissue as a tensillation of polygons. Like this picture is basically the wound that you see from the previous video, but it's colored by the actin. And yeah, we can define like each cells by these polygons. And so this is gonna allow us to study the shapes, the area, or like the interaction between the junctions. And so our model also based on the energy. So we incorporating like several physical terms. So the first one that I want to present is this called the elastic energy. So basically we incorporate the elastic strain energy at the subcellular level by triangulating like the cell or the polygon into basically multiple triangles here, depending on the centroids of the polygon. And then the equation for that is here. So the total elastic energy is the sum of the energy for each triangle. And each triangle, the elastic energy is representing by the Jg times A0 times W. The Jg is the volumetric variation due to the growth, and A0 is the reference area of the triangle, and W here is the elastic strain energy per triangle. The counterparts, because this one is representing in the discrete format, but it actually have the counterpart in the continuum model as well. So I want to emphasize on that. And then I want to talk about this elastic strain energy, because this one depends on the elastic coefficient or like the shear modulus. And we also have this called Fe, which is the elastic deformation. So I'm going to talk about the deformation tensor. So let's look at this diagram. I have like a triangle here. I call it like an initial triangle. And then after a while, it got deformed into the current triangle here. So the map such that explains like the explains the deformation of that is this so-called the deformation tensor F is basically the partial derivative of the current triangle X with respect to the initial triangle capital X here. And then we further decompose that tensor into two tensors, which is Fe and G, which is like elastic deformation tensor, and G is the growth deformation tensor. So the G deformation tensor is responsible for the stretching of the triangle. So we have like an intermediate triangle here so that is stretching in whatever direction the G gives. And then we have the Fe that store the stress and then bring that to the current configuration of the triangle here. So that the deformation tensor that I want to talk about. And then the next energy that I want to talk about is what is called the pressure energy. So this one is different from the previous one. This one acting for each polygon or the cells. And again, like this, the total pressure energy is the sum of the energy from each polygon. And so if you look at the equation here, the pressure energy per polygon depends on this capital K, the pressure coefficient, and then the A0, the preferred area of the polygon, and the A is the current area of the polygon. So you can see this equation as equation for the cells to have the resistance to the expansion or the compression. So basically, it's one like the area of the polygon to be about like the preferred area. And then we also have the assumption for the tissue growth, because we want to see the dynamics of the growing of the tissue. So here is the equation for that. It's basically an exponential growth for the preferred area. So the rate of that is depends on the gamma. And then we also introduce this what's called the tissue fluidity to the deformation tensor that I talked about in the previous slide. So the G here depends on the time. It has like two components. The first component is the isotropic part, which again depends on the gamma. It's the same gamma from the tissue growth equation here. And then we also have the rearrangement one that is modulated by the deviatory stress. And if you're curious about how I come to this equation, you can check my dissertation here. That's going to explain how like this equation is important, and it's also like energy dissipation and mass conservation that also like currently in like this equation. All right, and lastly, I have what's called the correlation energy. Is just to avoid, we just want to avoid like overlapping of the cells or like the unwanted collapse at the wound edges. So basically what we have here is we are looking at some a vertex here, and we're checking whether like it's getting close to, like too close to like the wound edges here or not. So if it's like within like some distance, which is like the D naught, then the energy is going to be there. So the system wouldn't want it to happen like that. So it's going to bounce back in the normal direction to that edge. All right. So once we have those energies, so we can compute the total force. So the total energy is basically the energy, all the energy of the one that I talk about, sum of them together. And then the force is basically the negative of the gradient of those total energy with respect to the position of the vertex R here. And so we want to try to solve like this equation to solve for the position of R such that they make like the total force to become to zeros. So basically we're trying to find like equilibrium solution to this system. And in order to do that, we can use like multiple software optimization, like the gradient descent method, or like the Newton method, or the accelerated version of that. In our current simulation, we mostly use this what's called the heavy ball method, which they involving with the kinetic energy and friction and mass to find like the optimized direction of the method so that we can find the equilibrium solution quicker. And so I want to mention about this what's called the Hessian of the energy, because this one is important. Like for example, you can use like this Hessian to tell you the optimal steps to use in the algorithm, such as the gradient descent. It's gonna tell you like how much of the small of the step size you need in order to have like a stable algorithm. And even like in the Newton method, you need like the whole Hessian, because you need to find the inverse of that and then to find the direction for that to reach the equilibrium for the system. And at the bottom here, I have the numerical algorithm for our simulations. So basically, we start with like step one, which is defining parameters or coefficients which we will obtain from experimental or like other models. And then we can give it like an initial configuration. And then the next step is we can employ like division process. But right now, unfortunately, in our current results, we not yet like included like that process yet. But in the future, we definitely gonna integrate that into our model. And then the next step, we're going to do the growth and fluidity. So basically, find a solution for the dA naught dt and dg dt. And then we can compute the force from like this equation and apply the software method so that we can have an equilibrium solution. That's going to be the configuration that we will see when the time is basically at the dt. And if you want to find like the sequence of the solution, of the time, we can like repeat the step 3 to step 6, like so on and so on. All right, so here is our results. So this is the wound closure simulation. So let's start with the left one here. So this is a wound closure. So I basically started with like a ring structure of the tissue. And then on the outer boundary here, I have them to be fixed, so it's not moving for the vertices. And then the color here is representing the negative pressure for each polygon. And interestingly, the front part of the wound when it's closing is more red. So basically, it means that it's not compressive. But the other like layers of the wound is more like compressive. And you can see there's some like that is like really blue area. That is what we call the buckling effect. And then in the middle one is the direction, like all the lines there is like the direction of the compression. So it's just telling you like in which direction that the cell or the polygon got compressed. And then the last one here is the color, the heat map color for the elongation. So basically, the brighter the green is telling you like how the more elongated of the polygon is. And you can see like this, the DT that I use in this simulation is about like 2 hours, because you can see that it's coming from like 2 hours to 4, 6, and so on to like 18 hours. Right, so that's like one result. Then we also try to study the parameter, the beta parameter, which is the rate of rearrangement. And so it's obvious that when you increase this beta, in the snapshot here is taking at like 10 hours. So when you increase that beta, the wound size is actually smaller, so indicating that the cell is basically less fluid. So when you're increasing the beta, so then the tissue is more fluid. And then when beta equal to 0, it's like more, it's less fluid. So this, the wound size is much larger compared to the other two. And another interesting thing about it is that the shape of the wound for the less fluid one is not really that circular. So it's kind of matched what we see in the MDCK video cells compared to like the one with like some beta, some positive beta, so they are more like circular. And the reason why it's more circular, it might be because of like, you can see the map here, which is telling you the elongation. You can see that when you have like larger beta here, you have more like green color, the bright green color around the wound area. So that might explain why it's get more like circular like wound area. So that our first application. And then we also have this, we are trying to use our model again to try to simulate this, what's called the growth and morphogenesis of the cell patches during a wing development of Adelphi. So we are trying to create this simulation from the experiment. So we asked our graduate student at the WPI, Yifan Gu, to create a model for, to create like a simple model for that. So basically we start with like a simple shape of the wing, and then we fix the vertices on the curved boundary, but we have the dynamic of the other side, which is like the straight line boundaries, to have the force only on the x direction, so it's only move in this direction. And so this is like basically at the end, at the frame 100, is an equilibrium solution at 1 hour, with some the gamma, the growth parameter to be like 0.55. So we are still like in the work of like try to analyze like this model to see whether it's like matched with the experiment or not. All right, so that's like all the result that we have. But I want to talk about the future work that we are going to do next. So first thing we are considering this what's called the neighbor exchanging procedure, like the T1, T2 transition. And we also consider this what's called the division, right? Because it should make sense to add that into the our simulation. So, but in order to do that, we need like a careful consideration of the energy, because we don't want the energy of our system to increase. We want to try to have the step, like when we're doing this like kind of thing, we want to make sure that energy is not increasing. And once we have that kind of thing figured out, we can continue working on like the wound crusher, trying to use our model to simulate like other type of the wound crusher and look at that shape and then compare that to the experiment, something like that. And then finally, we are also interested in using our model to also see whether it can match the experiment in the whole Drosophila notum as well. So if our model is working, then it's going to be impressive. And so I would like to thank our advisor, Minwoo, and also like the people in the Winmu labs at WPI, and also our collaborator, Shi-Wen lab at WPI, and also Yubing Sun lab at the UMass Amherst. And I also want to thank the organization, the organizer for giving us this opportunity. And of course, we would like to thank the NIH and NSF for supporting our work. And so now that's the end of my presentation. Thank you.
Great. Wonderful talk it was. I had just a curiosity that you told that you have not included the T1 transitions as in the normal vertex model, right?
Right. No, not yet.
So, I mean, in spite of that, do you see any kind of spontaneous neighbor exchanges in your simulations?
Yeah. So, like the problem, like for example, let's go to the simulation here. So you notice that because like we are trying to close the gap, so that means like the cell is enlarging. So, but like we have like a specific size or the area that it can grow.
Right.
So that means some of the junction has to get like shortened, and at some point it's like short enough that like basically the two vertices almost like at the same point. And
Correct.
so that's why like we actually like need a T1 transition to do the work. Yeah, to have like a stable kind of system.
Yeah. Oh, thank you. And another follow-up question is that you have the growth in the tissue. I mean, by a rate equation of the preferred area, right?
Yeah.
But is there any threshold that up to that threshold the area will grow and then it will stop, or it will grow continuously?
We grow simultaneously, but basically what we, in the algorithm, we solve it for a specific growth, and then we specify the growth at like dt time step, right? And then we solve for that solution, and then we move on and do that again, using the current the current growth that we have, and then so on and so on.
Yeah. Oh, so it will continue to grow.
With time. Yeah.
Okay.
It just depends on how long our simulation that we want to see.
I see. Thank you.
Thank you. Very nice talk. So you showed us these concepts for hexagons, hexagonal lattices. What happens if you take different shapes, triangles and so on? Would this change?
Yeah, it doesn't, well, actually it doesn't have to be hexagon. So it can be any polygons, like triangles, yes, for sure.
But do you get a different outcome if you change the shape, right? So do you find a different behavior or is it the same behavior?
For now, we are still working on that because we also do some refinement to our solution. So basically, maybe making it like more like less polygon, like making it into like triangles, right? And see whether that change our solution or not. So we are working on that as well.
Thank you.